Nesse caso, eles estavam utilizando LPS e concavalina A, que são 2 agentes imunoestimulantes, e aí eles estavam tentando identificar, dentro dos 115 analitos, quais a gente tinha diferentes quantidades quando a gente estimulava tanto com LPS quanto com concavalina A. E, por fim, eu falei, né, das BIDs, dos kits, e para a gente conseguir fazer análise desse tipo de resultado. Então, hoje, quem tem a plataforma, né, de aquisição citômetro, geralmente vai ter o software de análise, que nesse caso aqui, que eu estou mostrando para vocês, é o Belisa. Então, aqui, utilizando esse software, a gente consegue ver exatamente onde as nossas amostras caíram dentro da curva padrão. A curva aqui, geralmente a gente trabalha com 5 parâmetros logarítmicos ou 4 parâmetros logarítmicos. Isso faz com que a gente tenha aí uma amplitude muito maior. Então, a gente sabe que a amostra de plasma, a amostra biológica, a gente tem essa variação em diferentes condições, pacientes normais e pacientes com algum tipo de patologia, e isso faz com que a gente consiga identificar todo mundo, ou seja, eu consigo quantificar todas as minhas amostras justamente, porque aqui a gente trabalha com um limiar bem amplo. Muita coisa, eu vou deixar aqui meu contato de e-mail, então se vocês tiverem qualquer dúvida em relação aos kits, ou algum tipo de questionamento em relação à MERC, se vocês quiserem conversar, pode me mandar e-mail. Eu vou deixar o contato da Dani também, então se vocês precisarem de alguma cotação, ou se vocês quiserem fazer alguma reclamação, mentira, sem reclamação, gente. Vocês podem entrar em contato. Vou agradecer de novo a Sandra pela oportunidade, agradecer a vocês pela atenção e a disponibilidade. Obrigada. Dúvidas?
Perguntas,
Nenhuma?
pessoal? Eu tenho uma dúvida. Eu tenho lá meus macrófagos em cultura e eu quero deixar eles estocados para utilizar o seu kit. Como eu faço? Tem um reagente específico para fazer a lise?
Para a célula, o protocolo ele fica bem parecido com o de Western. Então, o pessoal acaba usando RIPA e algum inibidor de protease para fazer a lise, né? E também proteger, né? Nossa senhora, inibidor de protease. De maneira geral, a gente tem um protocolozinho para digestão de tecido, mas basicamente é isso. A gente usa um RIPA junto com o inibidor, faz a lise e deixa lá no menos 80.
Sobre o procedimento, o range eu vi que é bem amplo, né? Mas assim, um outro analito foge bastante. Aí isso muda um pouco aquele teu cálculo de quanto que vale a pena em relação à ELISA, porque se você tiver que repetir uma amostra diluída, você tem que repetir tudo de novo, né? Mas você tem alguma coisa, assim, dentre os analitos você está acostumado, isso tem acontecido ou tem conseguido tudo cair dentro da curva? Vou dar um exemplo, você acabou de falar com canavalina. Às vezes o interferon vai para nanograma em vez de picograma, e aí fica difícil pegar sem diluir numa cultura. E a segunda coisa é, só para ter uma estimativa, quanto que é uma placa com 80, que é que pega aqui, ou de 115 analitos, porque isso também, obviamente, que é vantajoso quando você pega por unidade, mas a gente não precisa de tanto, e aí o custo-benefício pode não ser tão vantajoso, só para...
Perfeito. Ótima pergunta, a sua também. Obrigada. De maneira geral, os kits, eles são testados e eles estão dentro de uma caixinha. A combinação dos kits, elas foram testadas. Então, você não pode usar um analito de um kit e misturar com outro, por exemplo. Então, assim, o que a gente testa? As diluições de amostra. De maneira geral, os nossos kits, eles são utilizados com 25 microlitros de amostra não diluída, soro plasma. Mas, obviamente, às vezes não funciona para tudo, né? Então, tem alguns casos, sim, de analito que fica abaixo do esperado, ou abaixo do limite de detecção, né, do meu ensaio. Isso acontece, infelizmente, em alguns tipos de ensaio. Então, realmente, né, eu tenho ali, não tenho TNF-alfa na minha amostra, tudo ficou muito maravilhoso para as outras, e para o TNF-alfa eu tive ali saindo fora do meu limite. A gente consegue extrapolar com esses softwares. Então, quando você muda o fit da curva padrão, isso, dá para pegar os valores, mas de maneira geral, né, eu não gosto de muitas extrapolações, porque aí eu acho que você perde também, né, todo ensaio ele tem o seu limite. Então, assim, de maneira geral, quando você tem abaixo do background, que a gente falou que é 0, eu considero como 0. De maneira geral, eu vejo caindo mais para baixo do que extrapolando. De maneira geral, o que eu vejo assim no meu dia a dia é indo um pouco mais para baixo do que para cima. Mas acontece. Aí eu falo, né? Claro, a gente pode fazer eu posso fazer uma prévia, posso fazer um piloto para entender um pouco a diluição, né? Como que ficaria. Obviamente que eu vou acabar gastando poço, mas dá para fazer esses testes também, diluindo ou não. Quando o kit tem uma recomendação de diluição, aí eu sempre falo, segue o que está ali, que eles já testaram. Dani? Pega o de 48. Faz o de 48. De 48 você não tem na cabeça, não, né? De cabeça eu não sei. Bom, é isso então. Obrigada, gente.
Deixa eu ver se ele acompanha aqui.
Às vezes é a versão do PowerPoint daqui, né?
E aí?
Good afternoon. Thank you very much, Professor Ricardo Gazinelli, for accept the invitation to present your work today here. I have a brief words to present you. Ricardo is a full professor of UFMG Research at the Fiocruz, Minas Gerais, visiting professor at University of Massachusetts in UMass, and also in the Faculdade de Medicina de Ribeirão Preto. He's a coordinator of INCT Vacinas, a member of a lot of scientific committees, Brazilian Academy of Science, the World Academy of Science, American Society of Microbiology, and also Brazilian Society of Immunology. Received a lot of awards, including the awards with your work with vaccines. I have a Leishtek vaccine for visceral leishmaniasis, a great work. Thank you very much.
Obrigado. Tem alguém que fala inglês aqui? Hein? Então tem que falar inglês, né? Tá, meu inglês está meio macarrônico ultimamente, mas... O slide está em português, né? Tá bom. Okay. So, first I'd like to thank the invitation. It's always a great pleasure to be here at the Department of Immunology, which I think is a fantastic department. It has trained lots of many great immunologists. So it's really, I think, very important department for immunology in Brazil. And also to see the friends. It's always a pleasure to be here and have some beer afterwards. And so I don't think I will talk about immunology. I'll talk about vaccine. And the other day there was the guy from Andrew Poller was visiting us, and he says, the last guy you need to make a vaccine is an immunologist. Of course, I disagree, because we learn a lot of immunology to get where we are. But I think this talk will be more like biochemist, and the idea here is just to give you an idea of what it takes to develop a vaccine. Because at the beginning we think, oh, I have a vaccine that works in mice, and that's go. Let's try in human. But it's very complex. So that's the idea of this talk, and that's what I've been doing on the last 2 or 3 years. So here is just to give you a perspective of our route during 2 decades. We began having a network of, it's called, it was the Minas Gerais network on biomolecules. Maybe Sergio Costa was part of it at the beginning. And here, the idea was to work on antigens from parasites that could be used as a target or diagnostic test, or even drug targets or vaccines. And so we started here. This was a group in Minas Gerais in the Department of Biochemistry and Immunology, where Sergio came from. And then we developed this vaccine that she mentioned as the Leishtec. And then in 2005, we became a national network that was the Millennium Institute, and now has a lot of people from São Paulo, Mauricio, Irene, a big group from São Paulo, from Rio. And then in 2008, we got renewed. And then in 2016, we got renewed again, and we have our physical lab that's in the Technological Park of Belo Horizonte. And here are some of the achievements. And then in 2000, and then we have the pandemic. And that we have a big jump. We got a lot of funding to grow. And that's when we are able to develop the first human vaccine. So I remember that during this period, I used to discuss a lot, and we used to be very frustrated because we protected a lot of mice, but we couldn't really never bring a vaccine to humans. And so despite all the terrible things that happened during the pandemic, that was when we really first had the opportunity to take a vaccine and go to a phase clinical trial. So that's what I'm going to talk about during my talk. Oops, estou fazendo alguma coisa errada? Devo estar, né? Eu apertei. Here. So this is actually were 2 articles published by I love the FAPESP journal. I received not the journal, but the electronic mail. And this was Carlos Fioravanti. Probably most of you know him. He's a famous reporter on scientific issues. And at the beginning of the pandemic, he wrote these 2 articles back to back. And And here is to gain time, we need to produce vaccine immediately. And here it talks about Butantan and Biomanguinhos that were ready to produce. They didn't have our own vaccine, but they make this tech transfer, and they, well, at the end, they were putting on the vial the vaccine. Oops. Oh. And then this part of the story is more related with why Brazil never developed a vaccine. Why? And that's what we got involved on that. At the time, the Ministry of Science and Technology opened a call to groups that wanted to develop a vaccine for COVID. There were about 16 groups, I think. And we were one among them. And we actually first, me and George Khalil, we had a grant together, and then they opened this call. And the big thing is, that's why I call the Vale da Morte. The Brazilian ecosystem of vaccines, I like to say that's almost complete. We had good teams working on vaccines at the university, in our institute as well. We have very good groups doing phase 3 clinical trials, and that's what we saw with CoronaVac and the Shadox AstraZeneca. We have the two big factories that produce vaccines for humans, and we have the SUS that have a big capillarity to distribute vaccines. So when we got vaccines enough vaccines, we got our population vaccinated very fast. The only problem is we don't have no one and no infrastructure available that could do this translation from the university to the patients or to the volunteers. And that's where we wanted to work on. And so I think there was a problem because we didn't have labs that produce vaccine in good manufacture conditions. We have no experience with phase 1 clinical trials. And again, we have another problem that's a very shy interaction between the industry and the universities. Opa, travou. Travou aí também? Tá. So into 2021, we had this meeting, working group from the Brazilian Academy of Science, and we wrote this short report saying how Brazil needs to have a group or groups that work on vaccine innovation. And one of the ideas, we make this available, but we listed the topics that we need to improve. But one of the ideas is that we would need to have centers that make vaccine, but research and development centers that take the ideas from the university and put them on a box of prototypes and bring to clinical trials and eventually to the industry. And that's our proposal. So we got funding from the Ministry of Science and Technology, SUFINEP, Governo de Minas, and FAPEMIG. And here is how the building will be, and here is where how it is right now. Today we have about 600 meters square of labs, and we plan to be about 8,000 meters. So the idea that we will expand our activities. So the first vaccine I will talk about is the COVID, is the one that's most advanced. And you all know about COVID. I don't need to talk about it. But the main point I want to make here, variants, and that's how the whole problem for the COVID-19 vaccine. They work beautifully if you have a vaccine to the right variant. But the problem is this: the variant emerge very fast and go down very fast. And as I was talking to Karina, when the vaccine is updated, so I think for 3, the bivalent vaccine for BA.1.4 and BA.1.5, but this strain was in 2022. And then in 2023, BB.5, but that strain was the year before. And now we are arriving with the JN1, which the peak is almost gone. So this is the problem that really, when we get the updated vaccines, so we have to deal with. And the idea, I think, more than the antibodies that protect against the severe disease. So again, I said that's the emergence of the variants is a problem. And then the other thing is the original antigenic original sin, and you're going to see in some of our results that even if you use the bivalent vaccine with 2 variants, the response that's induced against Wuhan is much higher. It's about 16 times higher. We have the data. I don't think I will show today. Then the neutralized antibody against the homologous variant. So, and then the other problem is the date of the vaccine. So at the time, we didn't have the notion of all that, but we said when we decided to make a vaccine for COVID, we said, no, we should do something different. We shouldn't do whatever Pfizer is doing and all the companies are doing because we have no chance. So what we decided is to take a protein that was very conserved, which is the nucleocapsid protein. By the way, Sergio has a vaccine that's very similar to ours. And there's a fusion protein with the RBD. Despite the fact that the RBD has lots of mutation, the T cell epitope seems to be preserved. And there are various papers showing that it's about 70 to 80% of the response to the variant is preserved in contrast to the neutralizing antibody that goes really down. So we decided then to make a vaccine for T cells, and the idea that if you have multiple epitopes, it's very difficult to have a mutation in all these epitopes at the same time that you will neutralize the T cell response. So this is the design of the vaccine, the nucleocapsid, that's highly conserved, and here the spike. And another reason we consider that's important to add the spike is because you have the T cell primed, active primed for the spike protein. So if there is upon infection, if there is a response, if you already have the T cells, this response will be much faster. So that's the second rationale to have the spike in the protein. So here are some of the papers. We published this paper. We use the adjuvant that was a poly-IC viral RNA, double-stranded RNA as adjuvant. And then we have a lot of work doing the combination of antigen and the adjuvant. And then we have the safety study with a different. We started with this, but in the middle of the way, we had to change the adjuvant because there is some issues about the heterogeneity of the RNA adjuvant, and we have to move to it's a squalene as a oil and water adjuvant. And then you have, ANVISA requires you do a lot of experiment to show the interaction, and then we have to do the safety test before going to clinical trial. So here is from the first paper showing the vaccine induces a strong CD4 and CD8 T cell response, and both in the lymphoid organs, but also at the resident. And I think here showing that the effector memory cells were the main cells in the memory cells are the main cells producing interferon gamma. And here is some of the anyway, yeah, it must be, these are different cytokines produced, and the most cytokines produced by the T cells were Th1. So suggesting that was a vaccine that induced a more potent T cell response. And this is the experiment. We wanted to show whether on this vaccine, because they induce high levels of antibody, but not neutralizing antibody. So we wanted to show whether the response was due to T cells or B cells. And here we use a B cell knockout too, and the mice stay resistant. But here we do the depletion. The mouse become partially susceptible. Both CD4 and CD8, they become very susceptible to infection. And this is contrasting with the control vaccine that was the AstraZeneca, where the response was primarily mediated by antibody. So this is the beginning, and that was the easy part. Well, we're very pleased that we plan to do a vaccine like that, and it worked. That was a celebration moment. But now we had the justification to go ahead and try to develop. And that part, we didn't know what we were doing, but we were lucky to get a lot of people from the pharmacy school that knew to work with formulations and lots of the physical, chemical, analytical tests. And the first thing we have, and here is some molecular biologists that work with proteins. So the first thing we have to do is to establish the whole process to produce the protein in good laboratory practice. And that is not easy because we spend a lot of time keeping the protein soluble, or you have to have a protein that's 99.99% pure, which is not an easy task. And so, and that was, we start producing this in bacteria, and then we cannot use nickel columns, so you'll have to use anion exchange column because you cannot use the polyhistidine. So we have to do this whole process, and then I get to the protein, and then we transfer to the lab that could produce in good manufacturing lab. The other thing that was very important, here is just while I'm talking, you can go and read all the tests we have to do to show that the protein was clean. And here is all the tests we have to do to show that the protein is stable. You have to do the stability test for 30, 90, 180, 270, and so on. But this is Graziella. When we didn't have an adjuvant, she went to all the patents of adjuvants, and she found one adjuvant that was no longer protected because the patent has expired. I see if there is people that work with vaccine here, it's called Adavax, which is this squalene. And we set the whole process to produce the squalene, and that was the adjuvant we use in our vaccine. So here is a highly pure protein. Here is a adjuvant. And then we say, where are we going to produce that? We went to the obvious place, but they didn't have space there. And then we got to Cristália. And Cristália, they were very generous. They say, we're going to do this for free. We just send your team here. We're going to set this for you. And we don't want anything on the patent. And that's our contribution to Brazilian science. And that's where the vaccine was produced. And we were very grateful to them. So next, I'm going to jump this fast because of the time. We did all the safety tests. That's when you have the formulation ready, you go to the safe. We show that the final formulation induces high levels of antibody, cytokines, and protect against the variants in hamster. And here is a Wuhan, here is the strain, and here is the Omicron. So we got a nice protection, and that was very important to approve the vaccine in Anvisa, because at the time you have all these variants, they didn't like vaccines that you couldn't change the spike. They wanted to have spike. In this vaccine, despite that we have only a small piece of spike, and we have the N protein, the vaccine has a good protective effect in both model. Hamster is a model of a more attenuated, moderate disease, and this transgenic mouse is a model of acute disease. You see, all the mice died, and the vaccinated were all protected. So and here is all the tests with safety tests. You do some biochemistry, these enzymes that for tissue damage, some for the inflammation markers, and so on. And at the end, the vaccine was very safe, and we got approved by the National Committee of Ethics and Anvisa to start the phase 1 clinical trial. And here is our vaccine. And here we did receive file a patent of the process and the final product. And here is when we start recruiting the patients, they got this, have the oxygen meter and so on to follow up. And here's the first patient that was immunized. He was a student of the PhD student at the microbiology department. And here's it was a big thing, so we had the minister, here's Elton, that does the coordinating clinical trial, Ana Paula, our dean, our rector, Sandra, and we had a big event. And here is the first thing, is the phase 1 clinical trial. We did try 3 different doses. That was 20, 60, and 100. And the first thing was to look for the side effects. The dark bar is the Covishield, which is the AstraZeneca. We have a little bit higher side effects, but I don't think this is statistically significant. We have only 9 patients per group. But anyway, our vaccine was shown safe, and we decided to take the 60 microgram per ml because this dose was sufficient to induce a high antibody response and a high T-cell response. So this was and again, we have a very strong T-cell response. Here is the protein of the vaccine, and was stronger than the people vaccinated with the AstraZeneca. So this was a good rationale. Then we got this vaccine that has the first studies were between people of 18 to 54, and here from 55 to 85. And again, we have a very nice response with the selected dose. So here it means like we are proud of it. I think it's probably the first example in Brazil of one vaccine that comes out of a concept and goes to complete a phase 2 clinical trial. And we did the whole process institution. So, and right now we have concluded the phase 1, phase 2. I wanted to show the results of the phase 2, but my Elton said that I couldn't, so next time I come. And then so we're in this phase that we're hoping to start the phase 3 early next year, but Anvisa has to authorize that. So the second, como é que está meu tempo aí? Tem 15 minutos. 15 minutos. Hein? Pouquinho não, muito. Né? Bom, eu vou falando aqui, se você quando faltar uns 10 minutos. 20 minutos. Tá bom. So the next vaccine I'll talk about is a vaccine. So the idea of this center is not to work only on our vaccine, but cooperate with other research institutions in Brazil. We have cooperation with USP, we have cooperation with Sérgio Costa. He started at UFMG, that's why we accepted him. But so that now there is no way we're going to get out of it, but we cooperate with him with vaccine for schisto. We also cooperate with startups. There's Immunoterra, where we work with this vaccine for HPV, and then a BCG, that's a company that has from a research at Butantan, that has also a vaccine. So our mission is try to help or to make it happen, right? It's not always. But this is the vaccine that's second most advanced, and that was developed by Irene. Irene is part of our National Institute of Science and Technology since the beginning, 2005. And her and Mauricio have developed this protein that has the epitope for B cells of the 3 main dominants of Plasmodium vivax. And here there is a region where there's a T cell epitope. And this, as I said, they have been working maybe for 10, 20 years. And now we saw the opportunity to take this to a clinical trial. So here is Irene Soares. She's a professor at School of Pharmacological Science. Ana Clara, Ana Luisa, and Santuza, the 3 of them work at the City Vaccines. And here is the first experiment. We generated this new formulation. We tested the same adjuvant, we tested our adjuvant, and we tested a combination of the RNA. And the reason we tested the RNA because it's a better inducer of T cell response. But at the end, we concluded that the idea, we came back to the old esqualene oil and water adjuvant in nanoemulsion. And here is a test of the vaccine showing that it induces a very strong antibody response, total IgG, ID1, using all adjuvants. But the reason why we decided to keep the same adjuvant was the same one because it was easy to approve in Anvisa. And again, we have a response to all 3 variants, a better response to the P-Vivax-like, but also to the VK210 and also to the VK247. So, and again, we have a nice T cell response, interferon gamma, maybe the Hiltonol, maybe a little bit better, the mixture. But at the end, we do have a nice response with our interferon gamma. And here is the most interesting result, which is the protective effect. We use this parasite that is a transgenic parasite that express the CSP from Plasmodium vivax. And this was done by a Brazilian that worked at the Pasteur Institute, Armino. Anyway, we see that the best protection we have was the one we chose, that was this our adjuvant with the protein, and here is the parasitemia, and here is the mortality. This is the list, the group that has most survival. So this further indicate that that was a good combination. The idea is that the protective response to malaria, to sporozoites, is mainly due to antibody. This slide is an example of a slide that you never should do one like this, because it's impossible for someone sitting there to follow. But I just want to, so we got, this protein was beautiful, and but the process they have developed, the protein got very easily degraded, and we have to repeat all again. So I'm going to go through all the process of getting I explained that on the previous for spin tech, but you have to get the protein, do the primary sequence, and you can see there is two bands here. We spent 2 months trying to explain why there is two bands until we found that there was for one reason, there was an extra amino acid in the protein that got cleaved, and the protein was produced in two different forms. But this is fine. As long as you explain why you have two bands, then Anvisa is happy. They're unhappy if you don't explain. And then we have to do the secondary structure, and then a tertiary structure where you use some fluorescence to measure the position of aromatic amino acids. And here you have to look at some translational modifications. And here primarily looking if there is any sulfate bridge. And so once this is done, you have to repeat this every month to show that the protein is stable. Again, so then we have you make to repeat 3 or 4 batches to show that each batch is exactly the same. You get mostly the same amount of protein, and so on, and you have to do all the purity and all that. You have really control of the process. And then you do the stability test where you have to do all these tests, again, the structure, the in vitro and in vivo. In vitro potency is antigenicity, in vivo potency is immunogenicity. That's bacteria, contaminant of the process, which are detergent and media to grow bacteria. Quite a lot. Use a diluted adjuvant, so that was considered as almost as the process of characterizing ability and so on. Finally, we got to the we had to do one thing that was important. The vaccine, the protein has to be lyophilized, because if it wasn't lyophilized, it degraded very fast. So we got the final formulation of this vaccine, which is the lyophilized protein and the adjuvant that was diluted one by one. And that's the way the vaccine will be used in the clinical trial. We're just about to submit that to Anvisa to have the clinical trial approved. And here's the patent that we submitted to UFMG and USP. And como que está o tempo aí? Mais 10 minutos. So now I'm going to be really fast. So we have a vaccine. I'm going to list a little bit of the projects we're currently doing. We have a vaccine for leishmania. She mentioned of the vaccine for dogs, and we use that as a basis for the human vaccine. And this is a work done by Bianca. In fact, what we did, we fused the A2 protein with a link cap, which is a protein from the leishmania mitochondria. And that's what we're using as a vaccine. We have went through all this process of characterizing the protein, all that. We have finished the safety test. Oops, here. So we are at this stage. We're basically at the same stage of the malaria vaccine. Here is the protein that we link up. We call it kappa 2 because it's link up plus A2. And here we're using the RNA adjuvant because leishmania, you really need to have a very strong T-cell mediated immunity. So we choose this. Here are some tests of potency, and here is the composition of the adjuvant. And I'm not going to go through this, but vaccine is a therapeutic vaccine, and it has a nice effect both in the hamster and in mice infected with Leishmania infantum. So it does work well. We have the proof of concept. We have the safety test. So here is the patent, and also this will be again a combination of two vials, lyophilized protein and the Hiltonol, which is suspension. We are working also in a vaccine for Chagas that, again, was initially developed by Mauricio Rodrigues. He identified the two antigens, ASP2 and transialidase. We made the adenovirus vector. Vaccine was beautiful, but without this problem of adenovirus, we decided to make a fusion protein. And so this is the protein we use for the vaccine. And in this, we are in the stage of establishing the process. We have in good laboratory practice, we just audit by the method. We have been working on a vaccine for monkeypox. In this case, we use the MVA, which is a modified Vaccinia Ankara virus. And that's it's a virus that grows in chicken fibroblasts, but doesn't grow in humans or mammals fibroblasts. And this is a vaccine that was not developed by us. This was developed by the NIH, was developed in Germany, and then NIH did a lot of work on that. And we received the virus from NIH, and we're scaling up the production and make in a way that it could be approved by a and then we are working again on RNA vaccines. We have RNA vaccine for dengue, and we have that uses the NS1, which is a non-structural protein 1, and E80, which is the target for the neutralizing antibody. And we have developed our own nanolipid particle, so we can get around the formulation from Moderna and Pfizer. And it's Renata and Gabriela, they're working on this. But this is show the distribution of the vaccine. This will have the nanoparticle with a luciferase protein, RNA, and also when you inject to see where the vaccine is going. Most of the formulation is going to the spleen and liver and stays for 72 hours. They have shown that it protects nicely. Here is using the DENV2, dengue virus 2. We have a good protection, and that's it. And no mortality protected animals are here. And now we are working on the tetravalent form, We have also worked on an RNA vaccine for H5N1. And again, it works beautifully. These studies were done with Edson Durigon here at USP. And here is the immune response antibody, interferon gamma, to the protein and to the virus extract. And here are the mice challenged with the virus intranasally. And you see that the immunized mice are totally protected, whereas the non-immunized are dead. And the thing here, we use this, again, it's a very conserved protein. It's not the target normally used for influenza vaccine. That's the hemagglutinin and the neuraminidase. And we have tested this vaccine by now with H1N1, which normally is influenza for swine, and 2 seasonal strains of influenza, and it did protect against all of them. I know people that work on the influenza hate that idea, but I think I like that idea. And we can discuss that later. And I think I'll jump, and that must be my last slide. So just to tell you the way we are organized, here is the way we are organized. We don't have a lab for each PI. We have a platform. And normally, the vaccines go. Some vaccines don't use all the platform, but here are the vaccines that we are working on. So we go step by step, and that's the way it works. We don't have and each platform has a leader, and so we have molecular biology, engineer of plasmids and virus, protein expression, regulatory office, guarantee of quality, and so on. So that's the way we organize, and each one of these are vaccines that we're developing. And here are some diagnostic tests that Ana Paula is working on. So here is part of the people that contribute to this work. This is our some Irene Santuza, elders helping us to put this on artificial intelligence and build some capability so we can be guided, maybe reach vaccine faster. Ana Paula Elton is the coordinator of the clinical trial. Gabriel also work with on this computational biology. Renan is our statistician. And here's a group of other associated researchers. And we now have about 75 people, 80 people in the institute. And normally the parties are great. And here are the funding agencies. And I think that's it.
Questions from students first?
I can start with, hi, Ricardo. It's absolutely awesome. I think you were a bit too humble when you told this story, because this is a story of a group of people led by you that brought basic science to vaccines, and vaccines in plural, as you just showed. This is a center that will develop vaccines in Brazil. This is the real first time, and I think it was clear that it takes more than a scientist. It takes a lot of politics and bureaucracies and regulatory and lawyers and things to get this going. I'm going to start with a very different questions, and this is regarding the structure of your center that is not related to labs run by PIs, but platforms, because we are here in the like a department still like academic, far from this. So how do you think we can, or is it possible to apply this structure to make a department program like postgrad program differently and more, successful?
Now, I like to say that this research and development science and the basic or fundamental science are two different cultures, and you shouldn't. They don't. Something that work for one not necessarily work for other. So I think we would never be able to do this if we were at least at the ICB in UFMG, because everybody thinks different. You want to do, so you do experiment and get the result, and tomorrow you want to do the next experiment. You don't want to wait. This type of research has to be very well planned. And it's much more expensive because all the equipments need to be certificated. It's a very high cost. And so I don't think we should, one doesn't apply to, I think like like we have here in most places now, you have the facilities. And that's a very good thing because no one can have his own equipment. In the past, it used to be that the guy had the equipment locked and I think that's one thing to have common facility. But I think the basic science is still something very artisanal, and you have to have your own world where you try your things, and I don't think it would work like this. On the other hand, here it's a team effort. So when I say there is 80 people working there, most people participate of the whole process. Some in the clinical trial, some on the preclinical, but there is a really, so it needs to be, and I can't have like everything set for doing one vaccine for the other. So that's the way I think it needs to be for this type of a more research and development science there.
Hey, Ricardo, just want to congratulate you. I pretty much follow the steps. We start together on the network, and you can show how you can, turn one academic project like networking biomolecules, right?
Right.
And become a corporate business of integrating basic science in translational science. So congratulations. I know how much effort has to put on this teamwork.
Thank you.
Hi, Ricardo. Thank you again. I'm kind of like seeing you quite a while in these meetings, and it's great to see more and more about everything you and all these people built. So congratulations. My question is like aligned with Gustavo. Is like, of course, you have in your team some PIs that came from the academia and that they have their own lab. But of course, that most of this work needs like people, hands-on people. So how is it the structure or the personnel recruited, because it's different, as you said, the culture is different. You cannot put like a master degree or PhD to make development of, like, I don't know, adjuvants. Can you, or I don't know, and do you hire people? And if you do, specifically for that, like, where does it come from, the money come from?
Well, we're very fortunate to get money during the pandemic. We got money from the Congress, from the mayor in Belo, and various grants from FINEP. The way it works, we have a committee that there are like 4 or 5 PIs. And normally we tend, like, for instance, I take care of the preclinical trial because that's what I know what to do, a little bit of the biochemist. Santuza takes care of biochemistry and molecular. But we discuss everything. We sit once a week, we sit and discuss. And each platform has a leader. Normally a leader of a platform is a postdoc or someone that has a more advanced scientist, but which a mind very focused on techniques and they take care of it. And then we have the people from each project that sometimes bring the sample for them to test. They have technicians, or they come and they do it themselves. For instance, for the preclinical test, each one come to the animal house and do its own experiment. But you have someone that, a leader on each platform. And in general, the platform leader, as far as I know, they're all post-level postdocs. They already have PhD, but some of the people that work in the labs are technicians with master or education. But something was very important. I was trained as immunologist, Santuza as a biochemist and molecular biologist, Ana Paula microbiologist. We didn't have much experience for the part of vaccine formulation, and that was, we actually were lucky to bring people from the pharmacy school, and they made the whole difference because they knew how to deal with Anvisa, they knew all the details of the process, not only, but all these analytical tests. So it's really a multidisciplinary group, and I know only a small part of it, and I think, that's it. I think that was one of the problems when we have all these projects that were funded by the Ministry of Science and Technology. A lot of people took this as a lab project, and this is not a lab project. It is a department project that has many expertise and so on. So, but that's basically, I don't know, but this is a very, yeah. Oops.
Thank you, Ricardo. Beautiful work and amazing. I work at Butantan, and we have this experience a little bit in the complex because we are commercializing some vaccines,
Right.
But I think it's very important to have an institute that develop the vaccines because the Butantan don't have this too much. They produce, they send, they sell, they have money, but it's very important. But the problem is what I'm saw is that when you do the phase 1 or the preclinical, we do the phase 1. Okay, the phase 1, that's true, more or less. But when we arrive at the phase 3, this is the problem.
Right.
Because sometimes you have to stop in the middle. That's what happens with the Butanvac. Because when you compare your vaccine with the Pfizer or the Moderna and on others, and then you are not at the same level, it does. You cannot produce and sell if you are not competing with the vaccines that you have here.
Right.
That is the problem. But I think it's very important, your work and then your project, your institution, to develop different kinds of vaccines that we can use with different adjuvants and all of this. Congratulations.
Yeah, I agree. Well, first of all, I have a big respect for Butantan, and I'm Phil Cruz and Bill Manguinhos. And that's something that always when people from Bill Manguinhos come to us and say, why do you? I think there is the two cultures that are different too. Is the R&D and the factories. And sometimes like the Bill Manguinhos, they try to do R&D with the regulation of factory, which makes it very difficult to develop a product, to do this type of research that you start to go to phase one, which is, I think my mission and our mission is innovation. I don't want to be a factory because I think if I become a factory, I'm going to stop doing innovation. And
Yes.
and that's my mission. As long, of course, we have a problem because we need to be funded because we don't make money, right? And that's something that we're gonna have find a way that we're fund to continue this work. But yeah, I think Brazil needs to do innovation in vaccine and diagnostic, and that's our idea. We are now working with Luciana on the BCG project, and but I think that's that's the thing. When you become a factory, you make so much money. You don't know. And if you go to it's a good example. If you go to the all or most COVID-19 vaccines, it's very interesting because they started at the university, and when like the Pfizer, they started the Mines University, they created BioNTech, and then Pfizer got, when the product was mostly ready.
Yes.
The Shadox, the Covishield, it was ready in Oxford, and then AstraZeneca came and got.
Yeah.
So I think there is this tendency that sometimes innovation now is not really, the factory don't want to spend time until they know that it's a good product. And I think that's so. I think we should have maybe more than one of those centers. But I totally agree with what you said. And phase three is a challenge, not only because of it needs to be better, that's fine, but the cost is very high, right? Yeah. We got the Pfizer is inducing terribly low levels of antibody when you look for the variant.
Yes.
When you look for the Wuhan, it's high, beautiful.
Yes.
But if you go to
The XBB, it's more or less.
and when we did for JNU, it was 16-fold less comparing to the Wuhan. And so I don't know if neutralizing antibody today is a good biomarker for COVID vaccine. And that's what we're going to discuss with some people. And I think also the influenza, we have the same problem, right? Some years the vaccine is good because you have the right variant, but some years it doesn't work well because the variant that's in the vaccine. So
And
I
then you
think,
take the variants that has most infected in the winter back in the US or in the Europe. That's the variants we choose to make the vaccine. And sometimes here it's not the same.
yeah, that's the one. So I think this needs a lot of discussion and maybe a different thinking.
True.
Anyway, we're having fun.
Yes.
Pessoal, se vocês tiverem mais questões, vão conversar com o professor Ricardo Gazinelli durante o coffee. A gente vai atrasar um pouquinho para iniciar os workshops, tá? Muito, muito obrigada, professor. Obrigada.